Yeah, let me share my screen. So one of the clarifications I just wanted to make, and I had intended to introduce them in the beginning, is that I manage the biomonitoring program for DEC. Meredith Streeter, who I believe is on the call, is the stream section chief. And then also on the call today, I believe, is Kelly Reynolds, who manages the WAVE program. And a lot of you have probably heard of the WAVE program and how it functions in the context of the monitoring program here. But I do appreciate when I talk to crowds, to your point, Russell, from the Lower Hudson, there's always, I think, the most interest from anywhere compared to anywhere across the state, in protecting their local water. So I really appreciate that, and certainly the turnout we see here today reflects that, because it's really impressive and always good to talk to such an interested crowd. So I guess I'll share my screen here and get started.
And my apologies for the introduction gaffes there, but thank you to the rest of your team who was all in attendance today. We really appreciate that.
Yeah, of course. So, when Emily asked me what I wanted to talk about, or, what I could talk about, I said, how about I, talk about ribs, but I really want to focus on the biomonitoring that we do, because aquatic insects and bugs are really cool. And, hopefully, how cool they are comes across in my talk, because it's been a lot of fun for me over the years, and hopefully you find something new and something interesting in what I would have to talk about today. So what I do want to talk about is the monitoring program background. So the RIBS program, both this touching on the lakes program, because we have kind of a sister program in the lakes section. And then certainly going into a little bit more detail for the flowing waters and the stream programs that we have. And then talking about biological monitoring using the aquatic insects, and how some examples of how we've applied those assessments of the biological condition. And then a little bit on data usage, how that information feeds into the process for data usage and regulations. And then lastly, as part of the RIBS program, we rotate around the state, and how to participate. So for all of you listening, if you have interest in learning more, participating in the process, whether it's through WAVE or, as we come in through the RIBS cycle next year, how to participate in that process, and then how to find data on streams of interest that we have on the web. So now, going back into the early 70s to give a little bit of context for the monitoring program that we have established today, it all really started with the 1972 amendments to the Clean Water Act, but those were driven by some of the environmental issues that you probably are familiar with. And here's a good quote from Nelson A. Rockefeller in 1965 on the Hudson River, right? So I'll read it real quick. The river from Troy to the south of Albany is one septic tank that has been rendered nearly useless for water supply, for swimming, or to support the rich fish life that once abounded. And so here's an image from the Lower Hudson River of a pretty nasty discharge. I believe this is actually an image from the Allegheny River here on the right, not the Hudson, but these were environmental issues that were prevalent at the time and really necessitated doing something about it. I think we've come a long ways on the Hudson River, but could certainly continue to improve. And then here's, because we have such a long history of monitoring in our stream section in the biomonitoring program, we have some good firsthand anecdotal evidence of the conditions that were present when we were out monitoring in the early 70s. And here's a good one that mentions on a sampler that we had deployed, grossly polluted, some things you can read here, white slime growing on the sampler. And then here's my favorite one, human hair wrapped around the bricks that were weighing down the sampler. So, we had staff out there in the early 70s that witnessed firsthand some of the gross pollution that was present in the Hudson River, which is not there now. So we've come a long ways and are continuing to work to improve the conditions of the Hudson and other water bodies throughout the state. So as I mentioned, the Federal Clean Water Act started it all, and the charge there was to restore and maintain the chemical, physical, and biological integrity of the nation's waters. That translated at the state level to environmental conservation law, saying that water shall be suitable for fish, shellfish, and wildlife propagation and survival. And I'm not going to get into it too much today, but if you want to read more about the classification, best usages of our waters, and the protection and how our data, applies from a regulatory perspective, you can read more about that in Part 701 in the classification of surface waters and groundwaters. So as I mentioned, I did want to touch on some of the other monitoring programs that we have. And across the state, we divide up our monitoring by water body type. So as I mentioned, I represent today the rivers and streams programs in the rivers and streams section, but then we also have a sister section, the lakes and ponds section. And then we have a groundwater program as well. That groundwater program is managed out of the Division of Water, but the sampling itself is actually done by the United States Geological Survey. So they sample the groundwater for us for DEC. So a little bit more about the programs. Within the rivers and streams section, we have the biological monitoring program, as I mentioned, Russell mentioned, I manage. We also have water chemistry sampling that's done alongside of that. And to a large extent, those are sort of within the stream section sister programs. They're done largely side by side at most of the sampling locations that we visit. And we also have the water assessment by volunteer evaluators, the WAVE program, which is the citizen science version of the biological monitoring that we do in the section. And it focuses on aquatic macroinvertebrates, aquatic insects, to evaluate water quality. And is a great program and feeds into the monitoring that we do. And then lastly, we have the toxicity testing program, and we do collect sediments and water column water samples for toxicity testing across the state. Within the lakes, kind of akin to our water chemistry sampling program, we have the lake classification and inventory program. That's the professional level program that the lakes section runs. And then they have the CSLAP program, or Citizen Statewide Lake Assessment Program, that is run by citizens like yourselves across the state, like associations that have been part of this process, hopefully, usually for a while. We have a long, rich history of the CSLAP program, and it really provides a lot of good data across the state with regards to several lakes. And then more recently, the harmful algal bloom program has really taken a lot of attention and really developed as HABs have been more and more prevalent and frequently blooming across the state. And I don't have a slide on it, but if you aren't aware, there is what we call the NIHABs application, and it's the way that the state can receive reports, or that you all can provide reports of potential harmful algal blooms that may be happening anywhere that you come across them, and this it's very helpful in tracking the prevalence and frequency of those blooms. So if you feel free to Google NIHABs, and you should be able to find more information about reporting of HABs. So a little bit about the rotating integrated basin studies cycle. So we operate on a 5-year schedule, and this is applicable to both streams and lakes, right? So a lot of this is very similar between our sections, with the exception of the WAVE program, which we kind of inserted here as year 0 as a lead up to our screening year in streams. But year 1, and for example, year 1, we're in year 1 in 2021, as you see here, for the Upper Hudson, Seneca, Oswego River Basins and Allegheny River Basin, is considered the year where we go out and we visit a large amount of sites within those basins and try to cover those basins spatially fairly densely, right? So we collect a fair amount of data at a lot of sites on a single visit. That goes for both rivers and for lakes. Year 2 has typically been our follow-up year to that. So we, based on the data we collect in year 1, we might go up and conduct more intensive data collection efforts in response to water quality issues that we've identified as part of the screening effort, or potentially developed other specialized studies that meet some objective for or data needs of other department programs, or clean water planning, for instance, anywhere that we need to collect data in support of some other program or need. And then year 3 to 5, and I have another slide coming up next, is consider our assessment cycle, updating of restoration and protection strategies, and completing what we call the water wheel, which I'll get onto next. And then ongoing, every year we have, from a streams perspective, we have the routine monitoring program. And those are sampled, those are sites, a network of sites across the state sampled quarterly. That's 4 times a year. We have the CSLAP program, as I mentioned. Those lakes are typically sampled every year. And then, of course, we have NIHABs, which is always active in taking reports of blooms across the state. The water wheel. So here's another look at basically at that 5-year rotating cycle that we're on, right? So after 5 years, we are coming back to the same basin. But I like to say, and I might be biased in this, that the water wheel begins and ends with monitoring. We conduct the monitoring, as I said, that feeds into assessment process. So that's how we take that data and digest it and use it from a regulatory perspective and assess our water bodies. The assessment of those water bodies then feeds into the planning and management strategies that are developed. And then based off of those strategies, they're then implemented through the development of clean water plans like total maximum daily loads, 9-element plans, SPEDIES permits. Those are the state pollution discharge elimination system permitting process to reduce contaminant loading to our water bodies to make sure that we're meeting designated uses and maintaining good water quality in the receiving waters. And then we have compliance and enforcement that is meant to, obviously make sure that those permits and limits are being met. And then we come back to monitoring, and we end with monitoring again, hopefully to show that the implementation process has been effective in improving water quality, or that's the hope at least. Doesn't always work perfectly or quickly, but that's the general idea of the division of water wheel. So some primary or program objectives that we have for both the Streams and Lakes program, the primary of which is the comprehensive assessment of water quality in support of the 305B and 303D lists. The 305B is our all waters list across the state. These are conducted on a bi-yearly or every 2 years. And then the 303D list is our impaired waters list. And those are the waters that are not meeting designated uses and in need of some sort of restoration strategy for improving water quality. We also have several secondary objectives, both within the Lakes and Streams programs. And the first of which is to identify and analyze temporal and spatial trends. And some of the monitoring that we do, I'll touch on how we do that. But looking at trends over time, as I said, we have a rich history of data dating back into the 70s. So we're looking at long-term trends from the 70s through the current day. We tend to put out long-term trend analyses every 10 years, and also spatial trends. So looking at conditions across the state spatially. We're also trying to characterize natural background conditions, looking at best attainable or least impacted conditions, tracking conditions, to the extent possible without anthropogenic influence, to look at the more subtle effects of changes like those induced by climate change. But also having that best attainable unimpacted condition allows us to modify and refine our sample collection and assessment methodologies to best reflect the evolving needs for better assessment and data in the assessment process. And then the third of our secondary objectives is to support the data needs of other department programs. So we're looking to support the regional offices that identify water quality issues and other core programs at DEC that are in need of information to support the work that they do. Like, as I mentioned, collecting information in support of watershed modeling, for instance, for a TMDL. So I mentioned the routine network from a Streams perspective. So now I'm going to focus more on Streams programs going forward and some of the details of them. The routine network is a network that is made up of 40 sites across a range of water body sizes and condition across the state to really get at the long-term trend conditions of our waters across the state. And these sites are sampled as we do with all our sampling, but representative, we take a very representative approach to sampling for the routine program. Equal width depth integrated water column chemistry collection. We're looking to also associate those collections with stream flow. So all of these sites are collected and co-located alongside of USGS stream gauge network. So we can couple concentration with stream flow to get loading over time. And as I mentioned, they're sampled quarterly every year. So regional staff are going out April, June, August, and October to sample these water bodies in order to get the seasonality over time. So the concentrations of parameters often change depending on what season we're in. So we try to capture that in the long-term data set. The idea here, of course, is to really get at that long-term trend and how water quality may have changed over time. Our screening network. So our screening network, we focus on collecting a lot of data, as I said, or a lot of sites and a fair amount of data, about 75 sites per basin for every year. So this is our draft site list right here for 2021 in the Upper Hudson, the Seneca, Oswego, and the Allegheny River basins. So we're looking to, like as I said, hit a bunch of sites. And we have historically focused on the macroinvertebrate community analysis as part of that, whereas with a routine network, it's a really water chemistry oriented type of sampling. We focus and put the most emphasis on the macroinvertebrate community analysis for the screening network. But alongside of that, as part of our efforts, we also include a brief habitat evaluation. We do collect information on sediment toxicity. We focus a little bit on recreational evaluation of those sites. And then more recently, we have also begun to collect water column chemistry with each of those samples. So we get a very good idea of what's going on in a fairly short amount of time. It usually takes us about 1 week to go out and evaluate an entire basin and collect those 75 sites for screening purposes. And then getting into the kind of special studies and surveys that we do, depending on the needs of the survey and the objectives of whatever we're setting out to do, we might select from our toolbox, right? So it can be any of the parameters that we would have collected as part of screening, but depending on the needs of the survey, we may collect different types of data to answer those objectives. So if it's, like I said earlier, a total maximum daily load or a TMDL, we might want to collect depth integrated water column chemistry and discharge to help inform a watershed model. If there's concerns about some kind of contamination issue, we could focus on sediment chemistry or macroinvertebrate tissue chemistry. So there's a lot of tools in our toolbox to choose from that we may employ depending on the needs of the survey, right? Pebble counts, fish community analysis is something that we focus on as well, bacteria, chlorophyll, algal toxicity. So it's a lot to choose from. So now a little bit more on the macroinvertebrates. So this is my favorite part and why we use macroinvertebrates, not only in New York, but nationally. They've really become very effective and efficient indicators of water quality over the last several decades because of their ubiquitousness and their relative ease and lack of expense when it comes to sampling, lack of expense when it with regards to something like water chemistry, which can be costly and you need to visit the site several times to get a good idea because chemistry can be very intermittent and change on a day-to-day basis. Invertebrates are efficient in that they reflect the conditions temporally much better than water chemistry, for instance. They're less mobile than fish. They don't swim away from influences or intermittent discharges, for instance. As a community, they're very sensitive to changes in water quality and pollution. So they're very diverse, and the macroinvertebrate community is shaped by the water quality changes that are seen in the river over time. And they're also easily communicated and perceived by the public. They're obviously macroscopic, they're big enough to see, and they're fairly easily communicated. And to sum it up, they're basically good indicators of overall integrated water quality over space and time. So when you collect a macroinvertebrate community sample, that is representative of the last several months of water quality, but also water quality throughout the basin. So that upstream contributing watershed contributes to shaping the invertebrate community that you're seeing at a point in the river. So now some more cool pictures on bugs. I used to do a lot of taxonomy identification of bugs, aquatic insects, and I took a lot of pictures as part of that, and I like to talk about them and show them. So these are our EPT, our Ephemeroptera, Plecoptera, Trichoptera, are typically considered sensitive taxa and oftentimes used as water quality indicators in and of themselves. So just briefly, I'm going to go through some of these organisms that I have photos of here. These are our mayflies here on the left. This is what we call a batisco, one of the not overly common, but really cool and very distinctive when you do collect one. Typically found in clean waters, obviously, but have these big, cool armored back plates that are very distinctive when you see them swimming around in a pan. Here, just to the right of that, is a heptagenaid mayfly, very flat and flattened dorso-ventrally, and do a really good job of holding on in high flow conditions. Have evolved over the years to do that, and effectively can withstand very high flows and rain events. And I have a cool video of a heptagenaid in a few slides here. And then down below that, we have a Euryalophella. It's another fairly, actually a little bit more tolerant, on the tolerant end when it comes to mayfly taxa. And then here in the middle are caddisflies. These are the Trichoptera. And this one, if you're not looking closely, it's called the helicocyche, and actually builds a case that looks like a snail shell. And if you get into the right water body, you'll see hundreds of these little critters in your pan. And at a glance, they look like snails, but they're actually, their cases are made up of sand grains that they've glued together in the shape of a snail shell, essentially. Really cool. And then we have a lot of really cool case building caddisflies. This is an Odontoceridae. They call these the strong case makers because, believe it or not, the cases are pretty strong. It's a very creative way of naming that group. But they are pretty tough to break. If you grab them with forceps, they are strongly made. So it is an accurate name. And then here we have a chimney case maker. These are filter feeding caddisflies. You see how their legs are oriented up. That's how they hold them in the water column when they build their case. They face up into the flow, and I have a cool video of this coming up. They basically catch particulates coming down through the water column and eat them. And then here on my left, this is kind of my personal favorite, the hydrocycid caddisflies. These are also filter feeding caddisflies, and they build their nets facing upstream into the flow of the river, and they're filtering out with their nets particulate organic matter as it comes down through the water column, and from time to time they come out and harvest their catch and eat it. So really cool. And one added piece that I think is really cool when it comes to the hydrocycids, we have several different species of hydrocycid, and depending on where you are, if you're in a headwater or if you're in a large river, the caddisflies build nets that have different sort of openings in them depending on where you are in that continuum of the river system. If you're in the headwaters, caddisflies like this build more open netting. If you're down in the lower reaches of the river, like the Hudson River, you will have a much finer net to catch the much finer particulate matter that's in the water column. So really cool. One of the reasons why I like bugs so much, and I guess I tend to geek out on them a little bit from time to time. And then here on the right, we have a odonate or dragonfly larvae, not actually a stonefly, but cool nonetheless. And then here are two stoneflies. Stoneflies tend to be predatory. So here are some of our more tolerant organisms. So on the one end, you have EPT sensitive taxa. On the other end, you have non-biting midges and worms that tend to be very tolerant of degraded water quality conditions. And when we see more of these types of organisms, we tend to be more concerned about water quality, and our metrics and assessments generally reflect that. So this is a midge that we caught downstream of a iron deposit, or sort of a leachate, where it was downstream of a landfill leachate actually, and we had a lot of iron in the stream, and hence the orange coloration of its little tube case. And here's how you identify midges. We mount them under high magnification on a slide and look at their mouth parts to really tell what organism we're looking at. And same thing with worms. They need to be mounted under a slide to identify them. Kind of very specialized taxonomy, a little bit harder, especially worms, I find very difficult to identify. You're looking at their chaetae, their little hairs sticking off their body, and the shape of the chaetae. So intricate taxonomy, tough, generally tough to do. So a little bit more about biomonitoring. I touched on some of the feeding methods of our macroinvertebrates throughout the river continuum, we call it. The river continuum concept this is a quote from the paper that initially introduced the river continuum concept, provides a framework for integrating predictable and observable biological features of lotic systems. So what does that really mean? It means that if you can identify where you are in the river continuum from upstream to downstream and set your expectations within that continuum, you can use that to your advantage to measure departure of the invertebrate community from that expected condition. And basically that's the basis from which we've conducted our biomonitoring over the years. So we have a set of biological metrics that we evaluate, and depending on how reduced those scores are of those metrics, we use that to evaluate the biological condition and water quality. So what this shows us here, and to touch on some of the feeding methods that I talked about earlier, is as you're in the upper headwaters of a watershed and you have heavy dense canopy and a lot of external material like leaf litter, twigs and stuff falling into the water body, you tend to have a lot of shredders and organisms that break down that material. A little bit of filtering of community, like those caddisflies that I talked about, collector gatherers, some predators, some grazers, but not so many grazers because you don't have a lot of primary productivity and light penetrating to the stream bed. As you move down into the middle reaches of the river continuum, you tend to have more collectors, definitely more grazers feeding on the primary productivity in the stream, and a lot less shredders, as you see here, the little tiny slice of the pie, far fewer shredders breaking down that, because there's not as much coarse particulate matter, not as many leaves falling into the stream as there were upstream. And then as you move downstream all the way to the bottom, you tend to have the majority collectors. So this would be like a river system such as the Hudson, where you have deep, slow, and much less benthic driven than you do up in the middle and upper reaches of your water body. But you see clearly that the functional feeding groups, these methods of feeding, are changing as a result of where you fall in that river continuum. And that's, as I said, the basis for biomonitoring and how we set our metric expectations for assessing biological condition. So here are some really cool videos I have that reflect the different feeding methods that I mentioned. This first one here, I'm going to play, hopefully that's showing up on your screen as well, is a caddisfly, a filter feeding caddisfly, a hydrocyclic caddisfly that spins its net facing upstream into the current. And you can see hopefully the net itself and how this hydrocyclic caddisfly is actually coming out and harvesting the particulate matter that has been captured by the net as it flows through. So really cool the way that happens, the way they do that. And as I said, the net size varies depending on where they fall on that river continuum, whether they're at the top of the continuum or at the bottom. Here is that chimney case maker that I showed earlier, the photo where the legs were sticking up. It's a chimney shaped case. They have themselves oriented up into the flow. And from time to time, they move their leg down to their mouth and eat the particulates that they've caught. And you can see it happening right here. And then lastly, we have a heptagenaid mayfly. So I mentioned heptagenaids and the mayflies and how they are very dorsal ventrally compressed. So they sit real close to rocks. And these are grazers. So he's going out and he's just scraping diatoms or rock, any kind of material growing on the rock, and eating it that way. So really cool, and a different way of feeding than the filter feeding organisms that you saw previously. So, I kind of touched on the river continuum, and the biomonitoring unit in the stream section covers water bodies that range from tiny headwaters to rivers like this, like the Niagara, some of you may have heard of, in the Hudson River. So we have methods that reflect those different conditions and expectations, depending on where you fall in the river continuum. For kick sampling in wadable streams and rivers, we use that in those upstream headwaters, the mid ranges of the river continuum, anywhere where it's wadable and we have a hard rocky bottom. So we use that for probably 90% of our sampling. We do multi-plate sampling and Ponar sampling for larger non-wadable rivers, depending on the type of survey that we're doing. For multi-plate sampling, we've been doing this for the last 40 or 50 years. And it's a way of us for tracking conditions of large rivers where they're too deep to sample So this is an artificial substrate that's colonized by the invertebrates over the course of a 5-week period during the summer. Ponar samples are for sampling the sediment dwelling community and soft bottom depositional rivers. And we use that from time to time, but by far and away, the kick sampling method makes up 90 to 95% of what we do. We are also continuing to develop new methods. So as I mentioned, we try to collect as many as we can unimpacted or least impacted samples and develop new methods to sample different niches within that river continuum. So low gradient conditions, as I'm showing here, are sort of water bodies and habitat types that don't fit the regular river continuum and change your natural expectations of what kind of organisms and what the community looks like as you move through that river continuum. So this is a methodology that we're developing and are in the process of finalizing now for sampling a different type of habitat. So we need a way of taking all of the organisms and their abundances that we collect at any given sample and converting it into something that we can use from a management perspective. So the first thing we do is take that community and convert it into a series of biological component metrics. So we are ultimately calculating an index of biological integrity, but it's based on several component metrics. And these are some examples of metrics that we use for our different methodologies. The highlighted or bolded ones are those that are relevant to the kick sample and used most commonly. But the biological metrics include things like species richness. How many different species do we see in a given sample? How many EPT taxa, so those sensitive taxa that I mentioned earlier, how many do we see in a sample? Hilsenhoff Biotic Index is a sensitivity index. So it's what is the community response to organic enrichment at a site? Organic enrichment, things like nutrients resulting in low dissolved oxygen, the community is going to respond to that. As I said, it's a very sensitive community. Percent model affinity. So the percent model affinity is an observed versus expected type of model. So we know what a community is supposed to look like absent human influence. So we are able to compare that model to the sample community and get a departure from that model in order to look at the percent model affinity. Species diversity. This is another component metric that we use depending on the methodology. Species dominance. What are the dominance of the top 3 organisms in a sample? And then non-chironomidae and Oligoceta richness is another one of our metrics that we may use depending on the methodology. So, different metrics are used depending on the methodology and where you fall in the river continuum. So we take all of those metrics, normalize them on a 10 scale, and calculate what we call a biological assessment profile score, or BAP score, which some of you may have heard of. This is, and we put the BAP scale on a 10 scale of water, BAP score on a 10 scale of water quality, I'm sorry. And what this figure shows is how the BAP score or the biological community condition responds to stressors. So what I'm showing here is what we can call the biological condition gradient. So just says as stressors increase, your condition is going to decrease. Stressors like temperature, dissolved oxygen, habitat degradation, increasing salinization of a water body. All these things help to decrease biological condition. And on that 10 scale of water quality that we calculate in the BAP, we divide it up into 4 impact categories. Non-impacted water quality is BAP scores of 7.5 or better. Slight impacted water quality, 5 to 7.5. Moderately impacted, 2.5 to 5, and severely impacted is less than 2.5. So these scores help us to assess water quality and measure whether or not the biological condition is meeting our impairment threshold of 5, right? So anything above 5 would be considered attaining our aquatic life use goals, or anything below would be considered impaired and potentially in need of some sort of restoration measure to improve water quality. So here's some examples of how we've used this data statewide. So here's a figure showing on the left 10 years of sampling, probably about 1,200 sites. If you convert all of those to BAP scores and use the color to represent those scores, you can get a quick glimpse of water quality across the state. And it tells gives you a lot of information very quickly just looking at it. You know, here's the capital district in Albany showing up largely as red because, you know, as we tend to urbanize, water quality tends to decrease. New York City goes down quite a bit. The Adirondacks and Catskills are largely blue, and some of our more agricultural regions out west are largely yellow and with some red, and it kind of reflects that degradation that you might see in some of those areas. Here's another example, a more site-specific or water body specific example of how we use BAP scores. This is actually the Susquehanna River out upstream and downstream of the Binghamton City wastewater treatment plant. After Hurricane Irene, we had a massive failure of that plant in 2011, 2012, and for quite a while, it was spilling raw sewage out into the Susquehanna River. And you see upstream of the facility a BAP score of 8 or 9. And then immediately downstream, you see that BAP score dip down into the 2.5 range, so severely to moderately impacted range. And you know, the reason was all that raw sewage was creating sewage fungus, and not a lot was living in those stressful conditions, not a lot of intolerant organisms anyways. And then as you move downstream another mile, you see the BAP increase a little bit. And then as you move another 6 or 7 miles downstream, the BAP is almost all the way recovered. And when you've moved about 12 miles downstream, the BAP score is almost back to where it was. You can see that biological recovery of the invertebrate community after about 12 miles. And I'm happy to say that we're going out this year to most likely document the improvement of the Susquehanna River downstream of this facility, because it's been restored and brought back online. And we're going to go out and hopefully show a straight line here of non-impacted water quality from upstream to downstream several years later. One more quick example. This one is pertinent to the Lower Hudson watershed. The Esopus Creek, after Tropical Storm Irene, we did several years of monitoring on Esopus Creek. This is upper Esopus Creek, upstream of the Ashokan Reservoir through 2011. And as I mentioned earlier, Tropical Storm Irene came through, dumped a ton of water, and we had just so happened to have sampled the upper Esopus Creek, several locations throughout the basin just before Irene. And we're able to capitalize on that and go out just after Irene. And you can see quite a dip in biological condition as a result of that scour. But as I alluded to earlier, these aquatic insects are highly evolved to survive that kind of catastrophic flooding, and are, you know, their methods of moving around the stream and kind of protecting themselves from that as a community are pretty well evolved to recovering from that. So we went back out a few months later, and things had improved quite a bit in only a couple of months. Densities were certainly down at this point, but the diversity of organisms had come back. And then a year later, conditions are essentially back to normal. So it's just a good example of the impact of a flood, but also of the resiliency of that community and bouncing back from it. So now briefly getting into how data are used and then how to participate. So all of this data that we collect is kind of digested through what we call a consolidated assessment and listing methodology. It's something that EPA requires as part of the Clean Water Act to help us assess and report on our water. So I mentioned the 305B and the 303D list earlier. We use this consolidated assessment listing methodology, or COM as we call it, to report on our waters and complete those listings in the 305B and 303D lists. This document outlines how data are used to determine the level of support and how we get to our assessments. And I mentioned it earlier, water body classifications have different best usages depending on that classification, and those best usages like drinking water, recreation, fishing determine how high or how good of water quality we expect on those sites. So that's a very brief and kind of high level summary. Hopefully it makes sense. If not, you know, we can talk about it later, or feel free to read on the COM. And as I said, part 701 of our regulations describes more on the water body classifications. So we're coming into the basin next year. So if you're interested in being part of the process, feel free to reach out to Kelly Reynolds as the wave coordinator, because in 2022, we will be conducting our screening of the basin. And we'll be holding a kickoff next year, right around the end of April, where we'll be talking about our plans for the basin for that year in monitoring. But if you want to participate, feel free to reach out to Kelly if you're not even already part of the WAVE program. It's quite easy to do. So WAVE, as I said, is a weightable assessment by volunteer evaluators, focuses on macroinvertebrate community sampling, and relies on citizen scientists to collect that information. You collect bugs. It's really cool, really fun, really easy to do. You send in voucher specimens so Kelly can verify the taxa that you've identified. And depending on what you found, right, it can take 1 to 3 hours stream side, pretty easy to do. But we're looking for a couple different conclusions, right? If you find what we call our most wanted macroinvertebrates, we can quickly and easily identify no known impacts. And actually that's very useful from our perspective in conducting our assessments to use that information from an assessment perspective. If you find what we call our least wanted macroinvertebrates, 4 or more of those, that goes on to our possibly impaired sites. And that's how it feeds into our screening year. We do set aside a certain amount of resources to follow up on possibly impaired sites that the WAVE program identifies for us. And then if you have another combination of macroinvertebrates, it falls into this gray area where we have no conclusion and don't really know clearly what's going on. But far and away, identifying high quality waters is really the most useful component of WAVE. So I encourage you, if you want to participate, I think a lot of people might focus on identifying impacts to water quality, but the most useful data that we receive from the WAVE program is these high quality waters that are identified. So as I mentioned, we're coming back to screening next year in the Lower Hudson. You see this RIBS figure here in 2022. So we're into the second column here of years. So we're going to have to remake this figure soon. 2022 is the Lower Hudson. So we're in year 0 right now for WAVE. So if you're interested in participating, feel free to do so if you're not already. And then lastly, I just want to leave you all with this. If you're interested in finding information on or biological data or HABs, toxicity, the fact sheets for a local water body near yourself, feel free to go and visit the New York State DEC Info Locator. This is the web link for it. Might not be a live link for you now, but if you Google the DEC Info Locator, it will take you to the site. And it contains all of this information: biological data, toxicity, HABs, the Whipple fact sheets, as we like to say. Those are summaries of the different segments of the PWL across the state. And the webpage looks something like this. It has all of our biological monitoring locations. You can click and add that layer. You can add air quality monitoring sites. There's more than just our water quality information, but you can kind of peruse through that. It's got information on how we calculate VAP scores, and we'll take you to the biological monitoring webpage if you want. So that's all I have today. Hopefully I didn't run through any of that too quickly, but again, I'm Brian Duffy. If you feel free to reach out if you have any questions afterwards and you can't talk about it today. And if you have questions on WAVE, how to participate, anything, feel free to reach out to Kelly. And she'd be happy to answer you. So I think that's it.
Great. Thank you so much, Brian, for that talk. And unsurprisingly, we have a lot of really great questions. So let's dig right into it. What is known about the biota of a tidal creek mouth? For example, how would someone monitor the health of the mouth of a tributary that is tidal and connected to the Hudson River?
That's a good question, and one that I think we run into from time to time when we get down into the Lower Hudson, but I don't have a good answer for a set of metrics or methodologies for doing that, because we really are focused on freshwater and those that are not tidally influenced. when we start to have that saltwater intrusion, the community changes, and we ourselves don't have good methods of evaluating that. So I'd actually have to dig in and and try to figure out what the best method to doing that would be, because actually historically we've monitored some areas that have some of that estuarine influence, and we get results that we don't know what to do with, and haven't gotten to the point of developing good assessment methods. And I guess the one thing I would say is that there are efforts going on now as part of, geez, I'm trying to remember the acronym for it, but there are efforts to develop assessment methods in those type of environments where you start to have the estuary, interface between salt and fresh water. So I'd have to send you information on that to get more into it. So I apologize, I don't have a good answer on that.
And Brian, would you expect the communities to also be different if it's freshwater tidal influence as opposed to saltwater tidal influence?
Yes, well, tidal maybe, but I guess I'm thinking more of the saltwater, the tidal, because the Hudson is tidal. I don't want to give the impression that we don't assess the Hudson River, but where you have that saltwater influence is where it really becomes critical. So it's not so much the tidal influence as it is the saltwater intrusion and the change. So somewhere, I think it's down around Poughkeepsie, we start to see that saltwater influence, and then our metrics and our assessments get all funky as you go down past that because of that influence. And you start to see the marine organisms showing up, and we don't know what to do then.
Sure. A lot of biomonitoring data shared through the DEC Info Locator is over a decade old. Is that the best place to find the most up-to-date information? How long does it typically take to publish that new data? And can you also speak to how the biomonitoring data feeds into the water body inventory or priority water bodies list assessment, if there's other data and factors that go into that water body assessment in addition to the biological data?
There's a couple questions packed into that one. We are working to generate a data portal that would provide accessibility more quickly to the biological data that we generate. It's something that we've been working on for a while, updating our process by which we make that data available, cleaning it up, because it's historically been maintained in kind of a disjointed way from a lot of the other information that we collect. So we are working on that data portal, and it's not quite ready for prime time yet, but hopefully we can provide that soon. But that's the short answer, is that we're working on making data more broadly available to everybody. And then the second part of that question, I believe, was how does this information feed into the water body inventory and other information? Well, the COM is the short answer to that. And that's the document that will provide much more detail on how this information, biological data, along with water chemistry data, is digested and put through into an assessment of a water body inventory or priority water bodies list segment. Did I answer that, or was that the same answer that I provided in the presentation? I don't know if I gave any detail.
I think that we can go back and look at the COM and follow up with you with additional, more detailed questions. I think that that would be. Do you receive or incorporate data that's collected in compliance with SPEDI's permit whole effluent toxicity WET monitoring requirements? The toxicity testing unit, that's Nikki Wright. She does that. She managed the toxicity testing unit, which is actually separate from the wet testing program, and she reviews that as part of the wet testing program. I don't personally look at those results, and they don't necessarily feed into water body assessment either. They're more specific to the SPEDI's program and the facilities that they're pertinent to. If you wanted more detail, we could certainly provide that, or there's information on the web, or Nikki is available to
answer questions because she's an encyclopedia of information regarding the wet testing program.
Perfect, thank you. I've got two questions here about wave and lakes. So can you do wave monitoring in lakes? And if not, is there a similar program under the lakes group for lakes that might feed into creeks or be part of a watershed?
One of the, you can't do wave monitoring in lakes. You know, one of the problems is that the macroinvertebrate, the bug community, is quite a bit different. We're actually, one of the things I left out of this is development of a lake biomonitoring method. And that's like low gradient sampling is in development, and hopefully in the next year or two should be something that we publish. But we don't have a citizen level biological component to that yet. You know, hopefully in the future, that's something we could develop. You know, the closest thing we have to citizen monitoring in lakes is sea slap. So, but we don't have the same biological element in lakes established like we do in streams with wave.
Great, thank you. How would someone find out expectations for their stream based on the river continuum concept and understand macroinvertebrates results compared to those expectations?
Our SOP, our standard operating procedure, describes the difference between what we consider a headwater and the rest of the wadable stream continuum. So if you wanted to and were interested, you could look up, or I could send it, but the standard operating procedure is available at our biomonitoring homepage. And, you know, depending on where you are in the state, the expectations of a headwater are slightly different, but they are dependent. A headwater is defined based on elevation and drainage area. So for depending on where you are, it could be anywhere from 6 to 10 square kilometers. I think that, I forget how many square miles that is, but smaller watersheds are headwaters, and they tend to be higher elevations, say above 1,200 feet in elevation. And there's a set of metrics that are associated with those sites. When you move downstream from there, you get into our more generally applicable statewide wadable stream assessment method and metrics that are associated with that. So all of those expectations are available in our standard operating procedure on the biomonitoring homepage.
Great, thank you. Probably just have time for one more question. How has the biomonitoring program evolved to address the idea of aquatic life use standards?
It is, that's a funny one. I wonder who asked that, because that's an insightful question based on some of the changes that have taken place over the last few years. And historically we have used the fishing use, right? So aquatic life is protected by the fishing use historically, or has been protected by the fishing use historically. So we have connected our biological assessments to protect the fishing use. You know, there's been some changing ways in which that has been applied in recent years. And you know, I guess at this point, I would say, you know, can look for look at the COM, and there the COM will provide good insight into the way the aquatic life use has evolved in our current thinking.
Great, thank you. So you've offered us a lot of the science behind the rationale and methods that DEC uses, and lots of more details in the consolidated assessment, listing methodology, the COM, and the standard operating procedures that biomonitoring unit uses, right?
Yes.
Great. Well, we're at time. Thank you so much, Brian, for coming and sharing. And if folks have more questions, feel free to follow up directly, because I know there's lots that we unfortunately didn't get to this morning. Oh, Russell, you're muted.
Yes, thanks, Brian, for a great presentation. For those of you in audience land, we do have a final speaker for this annual cycle next month. And don't forget for Toast to the Tribs. So thank you all, and we'll see you next month.